Carbohydrates are not our only source of energy. As we saw before, dietary protein is digested to amino acids that can also be used as an energy source, although this is not their major metabolic function. Most of the amino acids used for energy come from the diet. In fact, it is only under starvation conditions, when stored glycogen has been depleted, that the body begins to burn its own protein, for instance from muscle, as a fuel. The fate of the mixture of amino acids provided by digestion of protein depends upon the balance between the need for amino acids for biosynthesis and the need for cellular energy. Only those amino acids that are not needed for protein synthesis are eventually converted into citric acid cycle intermediates and used as fuel. The degradation of amino acids occurs primarily in the liver and takes place in two stages. Stage 1 is the removal of the alpha-amino group. Stage 2 is the degradation of the amino acid carbon skeleton. In land mammals, the amino group generally ends up in urea and is excreted in the urine. The carbon skeletons can be converted into a variety of compounds, including citric acid cycle intermediates pyruvate, acetyl coenzyme A, or acetoacetyl coenzyme A. The degradation of the carbon skeletons is summarized here. Deamination reactions and the fate of the carbon skeletons of amino acids are the focus of this section. The first stage of amino acid degradation, the removal of the alpha-amino group, is usually accomplished by a transamination reaction. Transaminases catalyze the transfer of an alpha-amino group from an alpha-amino acid to an alpha-keto acid. The alpha-amino group of a great many amino acids is transferred to alpha-ketoglutarate to produce the amino acid glutamate and a new keto acid. This glutamate family of transaminases is especially important because the alpha-keto acid corresponding to glutamate is alpha-ketoglutarate, a citric acid cycle intermediate. The glutamate transaminases thus provide a direct link between amino acid degradation and the citric acid cycle. Aspartate transaminase catalyzes the transfer of the alpha-amino group of aspartate to alpha-ketoglutarate, producing oxaloacetate and glutamate. Another important transaminase in mammalian tissues is alanine transaminase, which catalyzes the transfer of the alpha-amino group of alanine to alpha-ketoglutarate and produces pyruvate and glutamate. All of the more than 50 transaminases that have been discovered require the coenzyme pyridoxal phosphate. This coenzyme is derived from vitamin B6. The transamination reactions appear to be a simple transfer, but in reality, the reaction is much more complex. The transaminase binds the amino acid aspartate that's shown here in its active site. Then, the alpha-amino group of aspartate is transferred to pyridoxal phosphate, producing pyridoxamine phosphate and oxaloacetate. The amino group is then transferred to an alpha-keto acid, in this case, alpha-ketoglutarate, to produce the amino acid glutamate. In the next stage of amino acid degradation, ammonium ion is liberated from the glutamate formed by the transaminase. This breakdown of glutamate, catalyzed by the enzyme glutamate dehydrogenase, occurs as is shown here. This is an example of oxidative deamination, an oxidation reduction process in which NAD+ is reduced to NADH and the amino acid is deaminated. The carbon skeletons produced by these and other deamination reactions enter glycolysis or the citric acid cycle at many steps. For instance, we have seen that transamination converts aspartate to oxaloacetate and alanine to pyruvate. These carbon skeletons will be completely oxidized through these pathways, and the electrons harvested will be used to produce ATP. The positions at which the carbon skeletons of various amino acids enter the energy harvesting pathways are summarized here. A summary of the deamination of an alpha amino acid and the fate of the ammonium ion is shown here in this figure. Now in this video, we've looked at two specific amino acids. Of course, there are 20 amino acids, but we've chosen two specific cases. And the cases that we looked at were aspartate. So we looked at aspartate, and we also looked at alanine. Just these two. So these two specific cases. And the alanine or the aspartate combines with alpha-ketoglutarate. And when it does that in that transamination reaction, which is the removal of the amino group, we end up with glutamate and an alpha-keto acid. And the alpha-keto acids that are produced depends on the amino acid. Now from aspartate, we end up producing oxaloacetate. And of course, that can be oxidized in the citric acid cycle. And from alanine, we ended up making pyruvate. And of course, that also can be oxidized in the citric acid cycle. Now, in terms of that transamination, glutamate has a nitrogen. And that nitrogen is removed in oxidative deamination. That's what deamination means. It means removal of an amino group. So when that glutamate undergoes the deamination, it gets converted back into alpha-ketoglutarate. But in that process, not only do I liberate an ammonium ion, but I also reduce NAD plus to NADH. And what we're going to look at in a future video is what happens to this ammonium ion. And what happens is that it goes through the urea cycle and it gets excreted in the urine as urea. And you can see them here. So in this figure, you can see aspartate, which gets converted into oxaloacetate, and alanine, which gets converted into pyruvate. And then you can see how oxaloacetate and of course pyruvate can enter into the citric acid cycle and be fully oxidized. You don't have to have the fate of the other 18 amino acids memorized, but the summary of the degradation of their carbon skeletons is shown in this figure.